just to finish the presentation. Okay, let's finish it. I'm recording again, so this was the little conclusion. Now we are going to focus on the membrane peptides, which can be distinguished in two main groups, surface and integral membrane proteins. First of all, just mention the surface membrane peptides or proteins. Usually they have polar interactions or hydrophobic interactions. Types of them. The first group, which has the polar interactions, they have just the softer interaction with the plasma membrane, because just remember the main structure of plasma membrane. So inside there are the fatty acid chains, outside and inside there are the polar hats of the phospholipids. Yeah, this charged part. And the peptides, which has the polar interactions, there is only one way. They can interact only with the polar hat. Yeah, they are on the surface actually. Yeah, they have the interaction with polar hat, and therefore they can be easily dissociated. As an example, we can show you just cytochrome C. We can find cytochrome C in respiration in mitochondria, for example. The second group of the surface membrane peptides is the peptides which contain the hydrophobic chain. So it means that the interaction which is allowed can be only with the interspace of the plasma membrane, with the fatty acids which are inside the plasma membrane. So therefore, these peptides are incorporated into the membrane, plasma membrane, and therefore the bond is much stable. So the dissociation is not so easy. And usually the movement can go through the inner part of the membrane. Yeah, they are like buses which go through the plasma membrane. If we are talking about the integral membrane proteins, they have usually two characters. They have also the hydrophilic and hydrophobic part. They combine both principles. Usually they penetrate the whole plasma membrane. So they have the polar interaction with the phospho parts of the phospholipids, and they have also the non-polar interaction with the inner part of the plasma membrane. For example, they could be some hydrophobic alpha helixes, around 20 amino acids. They are placed inside the plasma membrane, and they are ended from both sides, or at least from one side, with something which has some polar interaction with the polar hat of phospholipids. So also additionally, there is some glycosylation effect. I'm talking about some sugar interaction, which is done by the association with oligosaccharides, and this oligosaccharide serves there as an active center for the recognition of some messenger, of some also immune system parts. So this is just the IP address. This is the QR code for our cells. Here at how the plasma membrane looks. This is quite nice model, talking about some fluidic mosaics. So it means that the plasma membrane, 2/3 of plasma membrane is created from phospholipids. It's these red dots with these orange tails. There are also cholesterol parts. That one, the yellow one. I'm not drawing. Okay. This yellow one. However, in this picture, there are just few cholesterols inside, but we know from analysis of plasma membrane that approximately 1/3 of plasma membrane content is created by cholesterol. So 1/3 cholesterol, and okay, cholesterol, and 2/3 as phospholipids. and the rest of it in range of percents, like 3% around, it's proteins. Yeah, they can go like alpha helix protein, maybe it's blue color, yeah, that one, alpha helix. They are on the surface, from the inner part, from the outer part, so surface proteins. Also, they penetrate the whole membrane, so talking about integral membrane proteins. And also they can create the channels, so also channels are created by proteins. And moreover, what's else there? I will use the green one. You will see these little green dots. So this is the sugars which can be attached to proteins as well as to lipids. And their main goal is usually the signalization QR code. Just remember that one. This is just an example of integral membrane protein. You may see that usually it's some complicated structure. In this case, this is rhodopsin, which is present in retina, and this protein is responsible for the visual perception. It recognizes the photons which are just dropping to our retina. So you can see that this part, which is just like this one, this is alpha helixes, 7 alpha helixes. They are immersed inside to the plasma membrane, and this part, these tails, are just flotating outside and inside the plasma membrane. Yeah, and these are sensitive. This is the sensitive parts for the attaching or attracting by photons, and the signal is transferred inside the cell via this complicated 7 body system to the cell. Then the chemical conformation is again changed inside the cell, and the chemical conformation initiate the perception of the light. So it seems to be that it's quite complicated. It is complicated because the information from the light is converted to the information to the chemical signal. And however, it's pretty nice just to imagine how all these information and signals are converted, how tiny is the barrier between the world of light, the world of chemicals, and our world, so what we perceive. So everything goes automatically without our sensitive contribution. We cannot control it too much. What about the membrane transporters? They are a little bit similar to channels. However, channels are like tubes. The transporters, they have little bit different shape. They are also inside the plasma membrane. They penetrate it, but they are open. They have 2 conformations. They are open from one side, then are closed. So 3 conformations, sorry. Then are closed with captured particle, and then open in the second part. Yeah, they just provide this movement, like open and capture, open and release. Yeah, this is just the switch movement, which is repetitively repeated. Usually it goes automatically without some support. Sometimes it requires energy. The difference with the protein channels, channel is open, channel is closed. This is the difference. ATP transporters. This is very well-known topic. You saw it also in the video. What's the time? Oh, here. Okay, we still have some time. ATP transporters, it means that they require some energy and they transport the particles against the concentration gradient. We also call them ion pumps. The energy which is used is the energy from the dephosphorylation of ATP, and also the majority of ATP transporters, they are also chemically, from the chemical point of view, it's alpha helixes. They usually contain different number of these alpha helixes domains. For example, ATP, sodium potassium ATPase, as you saw in the video, it has 10 alpha helixes, And the main aim of this ATP transporter is to pump sodium out of the membrane and potassium into the membrane. It helps to recover the resting membrane potential in general. Here you can see in the picture that normally we have, in the resting membrane state, we have sodium outside the cell and potassium inside the cell. However, after the action potential or because of some leaking, some amount of sodium can get into the cell and potassium can get outside the cell. So which is not really the state which is normal for the cell. So that's the reason that sodium potassium pump will just change and switch these two ions. So the aim of the sodium potassium pump is pumping sodium outside the cell and potassium inside the cell. However, because it is a proteins, and usually the function of all proteins like enzymes, channels, and so on within the body is really sensitive on some chemicals which can go outside, from outside, like in this case, digitoxin. So you know probably this plant, it's digitalis. In Czech is náprstník because of the shape of blooms. So you can put it into the finger, but be careful, it's really poison. A whole part of the plant is very poison. They are beautiful, but it's poison, and it blocks the function of sodium potassium pump. And if especially your heart is sensitive on that, and it can happen that some fibrillation or some diseases of heart can be caused because of the intoxication of digitoxins coming from digitalis. Okay, this is calcium ATPase. This one is present in our muscles, usually in endoplasmatic reticulum. The main aim is also the pumping of calcium from its low concentration to its high concentration, again switching for sodium. This is again for the recovery of because during the muscle contraction, calcium is released from endoplasmatic reticulum, and just for the recovery of the same state, we need to just push it back, and for this pushing back, we need some energy. So it's the same case like for sodium and potassium switching or similar one. And this one is also, this process is applied in digestion in our stomach, because in our stomach we have pepsin. It's an enzyme which is sensitive for the concentration of H plus atoms or ions, or HCl presence in general. So because of the presence of HCl, the pepsin is activated, and also by pumping outside and inside from the lumen, we can just coordinate the concentration of HCl in the stomach. So I think this one is too much for this course, so I'm going to skip it. And now I will just focus on the secondary transport. This is the last thing which I'm going to introduce you right now. And after that, you will have a time to fill the test. So in the second transport, the ATP energy is not used directly for the transport. Usually the ATP energy is used, the energy from ATP, the phosphorylation is used for the creation of some electrochemical gradient. Maybe, just an example, I will create an electrical gradient by pumping of protons from outside the cell or from the mitochondrial matrix to the intermembrane space. By this way, I create the electrochemical gradient. So I accumulate protons, H plus molecule, in one side, and then when the H plus molecule will be transported back, I will use the energy of the transport to transport or to provide some different transport. Yeah, this is just two-step reaction. I will show you an example. Just imagine a vehicle. However, the vehicle cannot just a car, okay, car. But the car, it's really on the manual movement. There is no fuel. So just to prepare on the situation, 20 years later. So we have a car without no fuel. However, we have to go with this car just to use it for transport. So we will push this car without fuel to the hill, and up to the hill we will leave it, and then down from the hill the car can go without our support. This is the same mechanism. So we will just accumulate the work for further utilization. An example is just the transport of glucose in intestine or kidneys, which you saw in the video. So here also in this picture. Now we have something like 20 minutes for your test. Just one question. Please look at your email. Do you have an email from me? I will send you the link also to the chat for sure. Okay. So you have the link on 2 places, in the chat and the email. So who has the link? Just raise a hand or tell me yes. Okay. CMN, do you have it as well? Katerina, yes. Mariam?
Yes.
Okay. So please open it. Maybe it's the best that you will open it right now. You have now exactly 20 minutes for the task. Just go through. This is just the your self-checking system that you remember the information which you just saw in the lecture. So there is all the questions are created from this lecture. It's there, and try to fill it. So I will check it after. So, okay. So go ahead, take your time.